the best of their beliefs. They are collages. No complete retrovirus nor its RNA in its entirety has ever been proved to exist either in vivo or in vitro. A further difficulty for the hypothesis was that it had never proved possible to show that the experimental observations attributed to retroviruses were exogenous to the cells used in experiments, i.e. that they came from outside of the cell. Indeed, all the evidence pointed to the opposite, i.e. that they were endogenous, inherent, to the cells themselves. Part of the evidence was that the so-called retroviral activity could only ever be induced experimentally in one type of cell, whereas HIV is supposed to infect many different types of cell in the body. The two contentions are clearly incompatible. The whole theory is rendered even more implausible when it is remembered that retroviral concentrations are always extremely low, which is why a huge excess of cellular material from patients is needed to be able to demonstrate any replicating virus at all. This, incidentally, is also the basis for the claim that HIV has only a low rate of infectivity. A much more rational explanation is that there is no virus there at all. History furnishes an unhappy precedent for this form of research. At the turn of the century, experiments were conducted using highly inbred laboratory animals. Under strictly circumscribed conditions, these displayed higher disease susceptibilities than animals which were not inbred. The phrase highly inbred was forgotten about, and generalizations about viral infectivity were made which turned out to be wrong, from which medicine has not recovered to this day. In like manner, experiments are nowadays performed with cell cultures instead of whole animals, for the very good reason that they greatly speed up experiments. The disadvantage is that this limits experimentation to just one of a few cell lines, which are always cancerous, because only they will grow continuously in the laboratory. History is repeating itself. Generalizations are made about the behavior of normal cells on the basis of results obtained from highly abnormal cells. Such cells can incorporate extraneous pieces of DNA into their own DNA when added to growth medium, as normal cells can too, only more slowly. Cells which have incorporated such DNA will obviously manifest characteristics for which the DNA coded, making it appear that a virus had been at work when nothing of the sort had happened. It is easy to see, therefore, how the bizarre notion of infectious DNA arose, and to conclude, wrongly, that a virus, in the conventional use of the word, is involved. The whole argument collapses, however, when it can be shown that non-viral DNA can be made to do this too, both in vivo and in vitro. If that DNA happens to be DNA arbitrarily defined to be HIV DNA or part thereof, then clearly the cell, which has incorporated the DNA, will behave as if it had been infected by HIV. Yet this is the basis of Duesberg's claim. In his retrovirological zeal, he does not seem to appreciate that infectious DNA is a contradiction in terms. For what else is a virus but that folded up DNA wrapped in a protein coat, so that the DNA can be transmitted from one cell to another, is what is normally called a virus. A loose strand of DNA could not do this by itself. It would be exposed to enzymic degradation. It would become entangled with other components. How would it identify its target? How would it get there? How would it enter it without a specific mechanism? A man of Duesberg's abilities should need no prompting that replicating equals cloning something in a test tube, and then detecting that something equals molecularly cloned DNA in a place where you have previously put it, is a circular argument, and therefore no argument at all. But then, tautologies are an indispensable part of all retrovirology, as I was at pains to point out when explaining the fallacy inherent in HIV antibody tests. Conclusion. The rules demonstrating the existence of HIV and retroviruses in general were never adhered to by those who devised them, nor were they ever validated. It is now easier to understand why people felt it necessary to inquire of this magazine what the rather self-evident term isolation meant. Suitable synonyms might be pure and or free of contaminants. They clearly had a slight worry at the back of their minds all along, that the term was used in retrovirology rather, as in Alice in Wonderland. It means what I say it means. Until AIDS was invented, retrovirologists were a minority sect who were happy to accept each other's flights of fancy without being too critical. They could fiddle around to their heart's content, safe in the knowledge that retroviruses were the least dangerous of all viruses. Well-meaning and credulous colleagues as well as aspiring virologists, journalists, and, through them, laymen were mesmerized by incomprehensible jargon into believing that the massive data on HIV and retroviruses somehow meant something. Each property relating to HIV and retroviruses generally can be shown to pertain to the cells used in the co-cultivation experiments. At no time have there ever been any credible grounds for thinking that these properties and components had anything to do with viruses in general, nor with HIV in particular. No particle of HIV has ever been obtained pure, free of contaminants, nor has a complete piece of HIV RNA or the transcribed DNA ever been proved to exist.